Our first SoftChalk lesson in Unit 1, Lecture 3 deals with the peptidoglycan cell wall in bacteria. Now, here's our fundamental statements and bullet points for this particular SoftChalk lesson. First of all, the vast majority of the domain bacteria have a rigid cell wall composed of peptidoglycan. The peptidoglycan cell wall surrounds the cytoplasmic membrane and functions to prevent osmotic lysis. Peptidoglycan is composed of interlocking chains of building blocks called peptidoglycan monomers. In order for bacteria to grow during binary fission, they have to synthesize new peptidoglycan monomers in the cytoplasm, transport those building blocks or monomers across the cytoplasmic membrane, put breaks in the existing cell wall so the monomers can be inserted, and then connect those monomers to the existing peptidoglycan and cross-link the rows and layers of peptidoglycan. Many of our antibiotics inhibit peptidoglycan synthesis of bacteria and lead to osmotic lysis of bacteria. Most bacteria can be placed in one of three groups based on their color after specific staining procedures performed. These are gram-positive, gram-negative, or acid-fast. And these staining reactions are due to fundamental differences in the bacterial cell wall. Gram-positive bacteria stain purple after performing a gram stain, while gram-negative bacteria stain pink after performing the gram stain. And finally, acid-fast bacteria stain red after acid-fast staining. And here's our detailed learning objectives for this SoftChalk lesson. And we're getting into a number of these double asterisk objectives where we have to know in depth and will be a common theme throughout the course. After this lesson, you should be able to state the three parts of a peptidoglycan monomer and state the function of peptidoglycan in bacteria. You should briefly describe how bacteria synthesize peptidoglycan, indicating the role of autolysins, bactoprenols, transglycosylases, and transpeptidases. Briefly describe how antibiotics such as penicillins, cephalosporins, and vancomycin affect bacteria and relate this to their cell wall synthesis. State what color gram-positive bacteria stain after gram staining. State what color gram-negative bacteria stain after gram staining. And state what color acid-fast bacteria stain after acid-fast staining. So with that brief look at the bullet points and our detailed learning objectives, let's get into the content for the peptidoglycan cell wall in bacteria. Now, the only bacteria that lack peptidoglycan and lack a cell wall are the mycoplasmas. These are some of the smallest bacteria, and the peptidoglycan cell wall, we're going to learn, helps prevent osmotic lysis under normal growth conditions, but the mycoplasmas maintain a fairly even pressure between the outside environment and the cytoplasm by actively pumping out sodium ions. So they spend a lot of their energy actually pumping out sodium ions to make the environment more suitable and preventing osmotic lysis. So as a result, they don't need the peptidoglycan cell wall. And the mycoplasmas are, the cytoplasmic membrane of the mycoplasmas are the only ones that have sterols in their membrane. But with that exception, pretty much all the other bacteria in the domain bacteria, with few exceptions, have a semi-rigid cell wall composed of this chemical called peptidoglycan. And again, that's just the name of the chemical substance that makes up the cell wall, just like cellulose is the name of the chemical compound that makes up a plant cell wall. Now, we're not dealing with the domain archaea here. They too have a cell wall, but their cell wall is distinct from peptidoglycan. So let's take a look at the function of peptidoglycan in bacteria. And quite simply, peptidoglycan prevents osmotic lysis. That is, they prevent bursting as a result of osmotic pressure from a hypotonic environment. So when we learned about osmosis back under the cytoplasmic membrane, we mentioned that bacteria concentrate nutrients, which are dissolved in water as solute through active transport. So as a result, the bacterium's cytoplasm is usually hypertonic to the surrounding environment. In other words, bacteria are normally living in a hypotonic environment, and their cytoplasm is hypertonic to that environment. Now remember, in a hypotonic environment, the net flow of water is into the bacterium. And without a strong cell wall, the bacteria would burst from osmotic pressure from the water flowing in. So this peptidoglycan is critical to most bacteria to prevent them from bursting due to osmotic pressure from water flowing into the cell, because they normally are living in a hypotonic environment. Now let's look at the structure and composition of peptidoglycan. So peptidoglycan is a vast polymer. It consists of interlocking chains of identical building blocks called peptidoglycan monomers. So a peptidoglycan monomer is a building block for peptidoglycan. Like an amino acid would be a building block for a protein. And the peptidoglycan monomer consists of two joined sugars, N-acetylglucosamine, which we usually call NAG, and N-acetylmuramic acid, which we call NAM. So those are two sugars that we always see in the peptidoglycan, those two joined together. And then coming off the NAM, or N-acetylmuramic acid, are 5 amino acids, or pentapeptide. And these building blocks are pretty much similar throughout gram-positive and gram-negative bacteria. There are some differences in the order of amino acids in the pentapeptide, but they're fairly similar throughout nature. So if we look at this illustration here in figure 1A and 1B, this represents a peptidoglycan monomer. And of course, you don't have to know the chemical structure. That doesn't mean anything unless you've had organic chemistry or biochemistry. But here's the basic idea. These are the two sugars that make up the monomer, NAG and NAM. And these are sometimes called amino sugars because they have these NH or amino groups associated with them, and those are connected together. And then coming off the NAM are 5 amino acids, or pentapeptide. So that is the basic building block for peptidoglycan, or the peptidoglycan monomer. And there's another one with a slightly different amino acid sequence. Now, these peptidoglycan monomers are synthesized in the cytosol of the bacterium. And once they're made inside the cell, they then have to attach to a carrier protein, a membrane carrier or transport protein called bactoprenol. And so these are transported across the membrane by active transport. Remember, the cell wall, the peptidoglycan, lies external to the membrane, and the peptidoglycan building blocks or monomers are made in the cytoplasm. So these have to be transported across the membrane towards the outside of the cell so they can be inserted into the peptidoglycan to grow the peptidoglycan during normal bacterial division and growth. And we're going to look at that whole process shortly. Now, once the peptidoglycan monomers are transported across the membrane, then they're going to be inserted into the existing peptidoglycan, and then everything's going to be linked back together again. So it's a fairly complex process we're going to be looking at, but one that's quite significant to us. So by adding new peptidoglycan monomers by means of glycosidic bonds, these are bonds that hook the sugars together, hook one monomer to the next monomer. This links the monomers into growing chains of peptidoglycan. And then the long sugar chains are then joined to one another by peptide crosslinks. So as we'll see a little bit later on, the cell wall is typically made up of many rows of peptidoglycan, sometimes many layers of peptidoglycan. And all of these rows and all of these layers become connected by peptide crosslinks. And what that does is kind of make the peptidoglycan kind of the equivalent of a molecular chain link fence that surrounds the bacterium. It's quite porous, but it's very strong. And if we go back up to figure 2A and 2B here, here we see an illustration of some peptidoglycan. Now here's our monomer again, NAG, NAM, with 5 amino acids or pentapeptide coming off the NAM. So these monomers, this would be one monomer right here, connects to the next monomer by a glycosidic bond, a bond that holds the sugars together, which connects to the next monomer, which connects to the next monomer. And so this connecting of monomer produces rows of sugars or chains of sugars. But the key to the cell wall being really strong is that every row, this would be the first row, this would be the second row, this would be the third row, every row is connected to every other row by these peptide crosslinks, where a sequence of amino acids hooks the peptide coming off of one NAM to the peptide coming off the next NAM. And by making the rows of sugars and then connecting every row and then connecting each layer of peptidoglycan, that becomes like a molecular chain link fence. And there's a little animation also on YouTube if you want to watch that. And there's a little self-check you can do to see how you're understanding the composition of peptidoglycan. Now, one of our major goals in this SoftChalk lesson is to learn how bacteria synthesize peptidoglycan, because we're going to be talking about peptidoglycan throughout the course, both in terms of control of bacteria, in terms of initiating body defenses, in terms of how bacteria harm the body, many other things. So remember, in order for bacteria to increase their size following binary fission, where one cell splits in two, the bacteria have to put links, have to break the links in their peptidoglycan so that new monomers can be inserted. And they also have to break the peptide cross links between the rows and between the layers of peptidoglycan. And then once the new monomers are inserted, all of that has to be resealed. So you can picture it like kind of adding bricks to a brick row, where a row of sugars would be a row of bricks, and the mortar between the rows would be the peptide crosslinks. But for that to grow, you'd have to add more bricks, more mortar, etc., or add links to the chain link fence. So there's a sequence of events that occurs here during kind of simultaneously during peptidoglycan synthesis. We're going to break it into several steps here. So first of all, we are going to look at the bacterial enzymes called autolysins. Lyse means to split, auto means self. So these are enzymes that actually break the glycosidic bonds between the peptidoglycan monomers of the existing peptidoglycan. And then other autolysins break the peptide crosslinks between the rows and between the layers. So to insert new building blocks, the row itself has to be broken so that new building blocks can be added to the row, and the cross links between the rows have to be broken again so that the new building blocks can be inserted. And that's done with enzymes called autolysins. So one group of autolysins breaks the glycosidic bonds. And these are the bonds between the peptidoglycan monomer. Glyco refers to sugar, so this is what's actually breaking the bond between one monomer and the next monomer in a row of peptidoglycan, in the existing peptidoglycan. And then another set of autolysins breaks the peptide cross bridges that link the rows together. And that allows a gap to occur where new building blocks can be inserted for cell growth. So our first animation here is illustrating that. And let's take a look at what happens with the autolysins and their role in the synthesis of peptidoglycan. So in this particular illustration, this represents a little bit of a gram-positive cell wall. The point is that this is the peptidoglycan here, and we're only showing it 2 layers thick. Here, so we see the monomers here, NAG, NAM, with amino acids coming off. Now the building block has 5 amino acids, but when the building blocks are inserted, one of the amino acids is cleaved off, so there's only 4 remaining, and that actually provides energy for the process. This is the space between the peptidoglycan cell wall and the membrane called periplasm. This is the cytoplasmic membrane, and up here on top is the cytoplasm of the bacteria. Now, when the building blocks are made in the cytoplasm here, they have to be attached to a transport protein called bactoprenol, which transports them across the membrane so they can be inserted. So those are the various parts we'll be looking at over the next series of animations. So the first thing that has to be done is that the bonds between the monomers in the existing peptidoglycan have to be broken so that new building blocks can be inserted, and the peptide crosslinks shown here have to be broken. So here we see 2 sets of autolysins. The one in green there is breaking the glycosidic bonds between the peptidoglycan monomers, and the other one broke the peptide crosslinks. So let's look at that again. This autolysin is going to break the glycosidic bonds between the monomers in the row, and this autolysin is going to break the peptide crosslinks that connect one row to another row and one layer to another layer. So let's watch them at work again. And what that does is create kind of the equivalent of a molecular gap where new building blocks can be inserted so the bacterium can grow. So that's the role of the autolysins. Break the glycosidic bonds between the monomers in a row and break the peptide crosslinks between the rows and layers. And that way new building blocks can be inserted. Now, meanwhile, the peptidoglycan monomers have to be synthesized in the cytosol. The cytosol is the fluid portion of the cytoplasm. And it's actually synthesized and attached to the transport protein bactoprenol. And the bactoprenols are going to transport the peptidoglycan monomers across the cytoplasmic membrane to insert them into the gap made by the autolysins in the existing peptidoglycan. So if we look at our next animation, we'll see how the peptidoglycan monomers are being synthesized. So again, there's the label we saw previously. And remember, the autolysins have created a gap by breaking the glycosidic bonds between the monomers and by breaking the peptide crosslinks. But while that's occurring, the bacterium is also making monomers in the cytoplasm. And so here's, they're partially made. The NAG has already been synthesized. The NAM has been synthesized. And these are energy compounds, kind of like ATP and ADP. This is UDP, but it has a high energy phosphate bond associated with it. So anyway, the first thing that has to happen is that 5 amino acids have to be attached to the NAM to form the pentapeptide right there. Then UDP provides energy to attach that to the bactoprenol, and then NAG is attached to the NAM, and the monomer is made. Bactoprenol then transports the monomer across the membrane, inserting it in the gap. And they're all recycled, so 5 amino acids attached to the another NAM, NAG attaches to the NAM, bactoprenol transports them across the membrane. And the bactoprenol is recycled, and as more and more monomers are made, they're attached to bactoprenols all around the bacterium, and it's transporting them across the membrane. Now, the bactoprenol doesn't actually leave the membrane like shown in the illustrations. That's just to show you kind of what it's doing. What it does is kind of flip from top to bottom and dump the monomers in. But it's a little easier to show it by showing the bactoprenol moving. So now we've inserted 2 new monomers in 2 rows of peptidoglycan here. But right now, of course, they're not attached to anything. It's very weak. So now a group of enzymes called transglycosylases. Remember, anything that ends in ase is an enzyme. And so this is going to form the sugar bonds, the bonds between the peptidoglycan monomers. The transglycosylase enzyme is going to insert and link the new peptidoglycan monomer into the existing peptidoglycan in the cell wall. And if we look at our next animation, we'll see the role of the transglycosylases in the process. So here we have the two peptidoglycan monomers that were synthesized in the cytoplasm and transported across the cytoplasmic membrane by the transport protein bactoprenol, but now they have to be inserted into the existing cell wall. So transglycosylases are enzymes that will hook one NAG to the next NAM, and that NAM to that NAG, that'll form the bonds between the existing peptidoglycan monomers in the existing peptidoglycan and the new monomer, as we see here. So transglycosylase forms the glycosidic bonds that hooks the new monomer to the existing peptidoglycan, and now each row is intact. But the wall is still somewhat weak because there's no cross-links between the rows here. So our next enzyme is called transpeptidase, because it's going to involve in producing peptide crosslinks. That's going to reform the peptide crosslinks between the rows and the layers of peptidoglycan to make the wall strong. So if we look at this animation, we'll see the role of the transpeptidases. So up to this point now, the gap was made in the wall. The monomers were synthesized in the cytoplasm, transported across the membrane by bactoprenol. Transglycosylase forms glycosidic bonds to hook the monomer into the existing peptidoglycan. So the final step is to form these peptide crosslinks that connect one row to another row via the NAMs. So here we see the transpeptidase coming in, and it forms those peptide crosslinks by hooking several amino acids together between the peptides coming off the MAM. And during that process, one of the 5 amino acids is cleaved off and provides energy for these bonds to form. And now at that point, the new monomer has been inserted and the cell walls have all been resealed. So keep in mind, of course, this is occurring in all the rows, all the layers around the bacterium as the bacterium grows following binary fission. So this final animation summarizes that whole process from beginning to end. So there's the labels of what's what again. So remember, autolysins are going to break the glycosidic bonds between the peptidoglycan monomers in the existing peptidoglycan to help create a gap in the wall to insert monomers, while other autolysins are going to break the peptide crosslinks between the rows and between the layers. So the glycosidic bonds are broken, the peptide crosslinks are broken, and that creates the equivalent of a molecular gap where a new building block can be inserted. Now, meanwhile, in the cytoplasm, peptidoglycan monomers are being made. So 5 amino acids will be attached to the NAM. The NAM will be attached to the bactoprenol, and then the NAG will be attached to the NAM to complete the synthesis of the peptidoglycan monomer. So there's the 5 amino acids added to the NAM. An energy compound hooks that to bactoprenol, and then NAG bonds to the NAM. Bactoprenol transports that monomer across the membrane and inserts it in the molecular gap. It doesn't connect them together, but it transports them across. And so this keeps happening over and over again. More and more peptidoglycan monomers are synthesized in the cytoplasm, transported across the cytoplasmic membrane, and insert into the gap created by the autolysins. So that has to be all resealed, of course. So in the next step, transglycosylases are going to form the glycosidic bonds that hook one monomer to the next monomer in the row. And then transpeptidase is going to form the peptide crosslinks between the rows and between the layers by taking a series of amino acids and connecting the peptides coming off the NAMs together. And that creates the production of what we call the molecular chain link fence that forms the peptidoglycan. So that is the process for bacterial cell wall synthesis. And again, this is a very strong, sturdy organic material. It's quite porous, but it's very strong because all the building blocks are hooked together. Each row is connected to every other row, and every layer is connected to every other layer. And there's a little self-check we can do then on that section on synthesis of peptidoglycan. Now, as we go through the bacterial anatomy, we're going to start relating this to why we need to learn this, how this relates, for example, to the infectious process of infectious diseases or a big picture of infectious disease. So a lot of our antimicrobial agents inhibit peptidoglycan synthesis in bacteria, causing osmotic lysis. So remember, the function of peptidoglycan is to prevent osmotic lysis. If the peptidoglycan is not synthesized properly or is made in a faulty manner, or everything isn't hooked together properly, then as water flows in, since they're living in a hypotonic environment, the bacteria would burst. So if the bacteria is actively growing, these are actually cytal antibiotics. These kill the bacteria because the bacteria burst. But the bacteria has to be actively dividing for that to occur. If the bacteria wasn't dividing, then of course they wouldn't be making peptidoglycan, so the antibiotics wouldn't have any effect until it started growing again. So remember, the autolysins are breaking the peptide crosslinks and the glycosidic bonds. And so at that point, you have a weakness in the cell wall. Transglycosylase enzymes are going to hook the monomers together to form a row, and then transpeptidase is going to cross-link the rows and layers to make the wall strong, as we just saw in the previous section. So if the bacteria makes a weak cell wall, it's going to lyse from osmotic pressure. And there's quite a few antibiotics that work this way, all of the various penicillins. Some of the different generic names of penicillins are listed there. The cephalosporins work this way. The carbapenems work this way. Monobactams work this way. Glycopeptides like vancomycin work this way. Now we're going to talk about this in much more detail in Unit 2 when we get to control of bacteria. But we want to start bringing the idea of why we're learning this and how this relates from the very beginning here. So if we look at the penicillins and cephalosporins, two major groups of antibiotics we depend upon, these antibiotics bind to the transpeptidase enzyme. They have a shape that fits a shape on transpeptidase. These transpeptidase enzymes are sometimes called penicillin binding proteins because that is what the penicillins actually bind to. And remember, transpeptidase is responsible for resealing the cell wall as new peptidoglycan monomers are added. They form the peptide crosslinks. So let's take a look at an animation here and show how penicillins would inhibit peptidoglycan synthesis. We'll get past the labeling here. Okay, so remember when we get to the point of transpeptidases, the autolysins have already split the cell wall, the row and the peptide crosslinks to create a gap. The monomers have been transported across the membrane. Transglycosylases have hooked the monomer into the existing peptidoglycan in each row and layer. So the final step that's supposed to happen now is that transpeptidase is supposed to form those peptide crosslinks between the rows and layers by connecting the peptides coming off of one NAM in one layer with the peptides coming off of another NAM. So that's the job of transpeptidase, and that's what finally reseals the wall and makes it strong. Well, here's what the penicillins do. they have a shape that fits part of the transpeptidase enzyme. And so once the drug penicillin binds to transpeptidase, the enzyme is no longer active. It can't form peptide crosslinks. So the enzyme that forms the peptide crosslinks is being inactivated as long as the drug's binding to it. Well, meanwhile, the autolysins keep splitting the cell wall. Monomers are being made, but no peptide crosslinks form. Eventually then, because they're living in a hypotonic environment, as water flows into the bacterium, that causes the bacteria to burst because they have a weak cell wall. And we'll see later on that vancomycin, cephalosporins, bacitracin, and a few other antibiotics work that way. That'll be taken up in Unit 2 under control of bacteria. And there's a little self-check you can do there. Now, we can kind of divide bacteria into 3 groups based on staining reactions, how they stain with certain staining procedures. The most widely used stain in bacteriology is the Gram stain, developed by Christian Gram. And we'll be doing the Gram stain beginning in Lab 6 a little bit later on in the semester. But the 3 groups we call gram-positive, gram-negative, and acid-fast. Now the Gram stain, like I said, is the most commonly stained procedure used in bacteriology because it divides bacteria into 2 groups, gram-positive or gram-negatives, named after Christian Gram. The positive and negative has nothing to do with charge. They could have said gram 1, gram 2, gram A, gram B, or whatever. But here's the difference. Gram-positive bacteria retain the first dye in the Gram stain procedure, which is a purple dye called crystal violet. And because they retain that dye, they wind up appearing purple when observed through the microscope. And there's many, many gram-positive bacteria. Some of them we'll be dealing with include Streptococcus pyogenes that causes strep throat, the pneumococcus or Streptococcus pneumoniae, Staphylococcus aureus, common cause of abscesses, Enterococcus faecalis, Clostridium species, and such. And this is a photomicrograph of Staphylococcus aureus, where we can see they have stained purple in the Gram stain or violet, indicating they are gram-positive. Now, some bacteria with the same exact 4-step staining procedure that we'll learn in Lab 6 are gram-negative. And gram-negative bacteria decolorize during the Gram staining procedure. They lose the crystal violet iodine part of the Gram stain and become unstained, so they pick up a second stain, a counter stain called safranin, which is pink in color. And so since the gram-negatives decolorize and lose the crystal violet, they pick up the pink counter stain and they appear pink in a Gram stain. And we'll be looking at a lot of medically important gram-negatives through the semester, including Salmonella species, Shigella, the Gonococcus, Neisseria gonorrhoeae, the Meningococcus, Neisseria meningitidis, Haemophilus influenzae, E. coli, Klebsiella pneumonia, Proteus, Pseudomonas, and a bunch of others. So here's a Gram stain of E. coli in figure 10, and you see the gram-negative or pink bacilli. And this is a Gram stain I made of a mixture of Staphylococcus aureus, which is gram-positive, and E. coli, which is gram-negative. So we'll be doing a number of these in lab. Now the Gram stain is effective against for many bacteria, but one of the bacteria at the time of Christian Gram that they were trying to identify was Mycobacterium tuberculosis. But the Mycobacterium did not stain well in a Gram stain. Sometimes it didn't stain at all, sometimes it was variable, sometimes it looked weakly gram-positive. So eventually, a number of bacteriologists came up with a stain called the acid-fast stain. Now, the acid-fast bacteria, when stained with an acid-fast stain, which is different from the Gram stain, resist decolorization with an acid-alcohol mixture. And the combination of acid and alcohol combined is a very powerful decolorizing agent, much more powerful than the one used in the Gram stain. So during the acid-fast stain procedure, the acid-fast bacteria resist decolorization, and they retain the first dye in the acid-fast stain called carbofuchsin, which is a fuchsia or reddish color. And so they tend to appear reddish under the microscope. And for our purposes, the main acid-fast bacteria we'll worry about is the genus Mycobacterium, which includes Mycobacterium tuberculosis that causes tuberculosis. And figure 12 shows an acid-fast stain of sputum, and this is from a person that has tuberculosis. Now, virtually all other bacteria other than acid-fast bacteria lose that reddish or fuchsia-colored color. This is the Mycobacterium tuberculosis and acid-fast bacillus that's reddish there. These are some other bacteria present, and they've decolorized and picked up the blue counter stain. Epithelial cells, mucus, white blood cells, all of those decolorize and pick up the blue counter stain. So only the acid-fast bacteria appear a reddish or fuchsia color. And there's a little self-check you can do on that. And finally, at the end of the SoftChalk lesson is a self-quiz for the entire peptidoglycan and peptidoglycan synthesis SoftChalk lesson with some identify activities, etc. So make sure you go through that. Now, in our next 3 SoftChalk lessons, we're going to be looking at each of these 3 cell walls we just mentioned and compare them: the gram-positive cell wall, the gram-negative cell wall, and the acid-fast cell wall.